welcome to my channel. In this live of today, we will be learning how to draw diagrams of plant tissues. This video is being done for class 9 science NCERT chapter 6 tissues. This book is being followed in the CBSE board. So in case you are interested in learning how to draw all the diagrams in an easy way, you can definitely go ahead with this live. I hope the screen is visible to everybody. So let's begin with the plant tissues part from the chapter 6 tissues of class 9 science. So before I begin for the plant tissues, you must know that plant tissues, they are of two types according to the book, meristematic and the permanent tissues. As I'll speak, I'll talk about them as well. So meristematic tissues are the ones that continuously divide. So they keep on dividing and form new cells. We as a human don't have such type of meristematic tissues. So meristematic tissues are of three types if we talk about their position. So on the basis of position, they are of three types. First one is apical meristematic tissue. Then we have intercalary meristematic tissue. So empty here means meristematic tissue. And then we have lateral meristematic tissue. Okay, so we will be learning the diagrams of all the three types of meristematic tissues because we are learning that as per the position they are of three types, so where they are being located. So let's go ahead and then we will be drawing the diagram. So suppose if this is the plant and these are the branches. So this chapter must be coming in half yearly. You must go ahead with this diagram because most of the times it is being asked. So I'm using different colors so that it should be clearer to you. And here at the tip, these are apical meristematic tissues. So apical meristematic tissues will be always there at the apex, right? So this is apical meristematic tissue which is present at the apex of root or shoot. Because I have drawn branches here, so this is actually the stem. Now see, this is the branch, right? This is another branch here. I should color it with green so that there should be no confusion to you. So this is the branch and this is also the branch. Now the place where the branching occurs, right? That position in a stem is known as node. So at the base of the node means the area where I am coloring it with black. This place also, because if you see along its side, the branches are seen. So this is also the node. So at the base of node, what do we have? We have intercalary meristematic tissue. Intercalary meristematic tissue. If a question comes to draw, definitely draw it. We will talk more about it, but let's learn the location. The place where I am marking it right now, this is lateral meristematic tissue. So apical is present at the apex, intercalary is present at the base of the node, and lateral is present along the lateral line. Just to brief you about these meristematic tissues, apical and intercalary, they will always increase the length of the plant. So they are causing longitudinal growth or the primary growth. Why are we saying primary? Because this is the type of growth that is occurring first in a plant. Also, lateral meristematic tissues are only present in the dicots. Di means two. Cots means cotyledons. So you might have seen rajma having two cotyledons, gram having two cotyledons, mango inside the stony part has two cotyledons. So all the plants that have two cotyledons in them, they belong to a category dicots. You might have seen that mango tree over a period of time it increases its diameter. Why? Because it contains lateral meristematic tissue. So lateral meristematic tissue, they help in attaining the secondary growth. Why are we using the word secondary? Because once primary growth will be done, then only secondary will happen, and this will increase the diameter of the plant, right? So this growth is increasing the diameter. In the NCRT, you may also find the word girth. So girth is also important. In case you want to write it down, you can write this as well. So if a diagram comes on the meristematic tissues, please make sure that you use colorful pencils because it is home examination and nobody can deny from this. So we are done with the meristematic tissues. Now, before going ahead, let me brief you that permanent tissues are of two types. And first, we will be going ahead with simple permanent tissue. Simple permanent tissue in plants includes parenchyma, collenchyma, and sclerenchyma. Scleren. For ninth class students, it must be a difficult uh, task to pronounce all these words, but first you should learn the pronunciation. So these are pronounced as parenchyma, collenchyma, sclerenchyma, and they come under the category of simple permanent tissue. Now, if I brief you, they are the living ones, parenchyma and collenchyma. This one is the dead tissue. So, dead tissue, what is the use of it? It will be adding mechanical strength to the plant. So because of this strong mechanical strength, there are certain plants that are able to bear strong storms. But there are certain plants which, when faced with high storm conditions, they lie flat on the ground, right? Because they have less sclerenchyma in them. So sclerenchyma adds mechanical strength. The more the sclerenchyma, the greater the mechanical strength. The lesser the sclerenchyma, the lesser will be the mechanical strength. Now you might have seen that leaves they bend according to the direction of wind. Why? Because they have collenchyma in them. And what does collenchyma do? It adds flexibility to the plant so that the plant can move without actually being breaking down. Parenchyma is the living tissue. Most of the activities, normal physiological activities are done because of it. So parenchyma is a simple tissue which is present everywhere in the plant. Let's see how can we draw the diagram of parenchyma. Let's begin first with the parenchyma. So in case you cut the transverse section of parenchyma, you will be able to see cells like these. While making the cells, you need to be careful. Please make the end point of each circle neatly. See, I am trying to cover each cell in a definite way because end to end connection is there between the cells. So this is how a parenchyma cells can be drawn. And if you see this area in between where I am putting a red mark, right? This is intercellular space. So whenever in a transverse section or a cross section, what is transverse section or cross section? सो आई होप एवरीबडी ईट्स क्यूकुम्बर। जब आप क्यूकुम्बर को काटते हो, राइट, सपोज दिस इज द क्यूकुम्बर, आप कट करते हो लाइक दिस इज ट्रांसवर्स प्लेन, तो आपको क्यूकुम्बर का साइज कैसे मिलता है? राउंड। सो इसीलिए जिस भी टिश्यू में पेरेन्काइमा इज देयर, इफ यू आर कटिंग इट, you will be able to see intercellular space, right? Intercellular space means the tissue is parenchyma. What is parenchyma? It is a simple permanent tissue. When we will come to the explanatory videos, then I'll explain you what are the various functions of parenchyma. Let's go ahead and begin with another simple permanent tissue, but this is going to be collenchyma this time. So we will be making collenchyma now. For making the collenchyma, you will be making hexagonal cells like this. You will make vacuole in between like this. The teacher just wants to see whether you know or the labels or you don't know. And then we will be making thickening. So while making thickening, please make cells like this. So this is the thickening that you can see. So thickening is present in the form of angles, and the thickening material in the case of collenchyma is pectocellulose. So thickening in the case of collenchyma is pectocellulose, which is made up of pectin plus cellulose. And they are the living tissues in plant. So this is the thickening in case you want to label. This is the thickening. This is the cell wall. This is the vacuole portion. Okay. This is the vacuole portion in the center. So these all labelings are enough for class 9 student. So this is collenchyma, the second type of simple permanent tissue. Now let's begin with sclerenchyma. Sclerenchyma. Sclerenchyma is a simple permanent tissue which is dead in nature. Let's see how to draw this. So you will again be making hexagonal cells, right? So these are our hexagon cells and we need to make thickening around them. So for making the thickening, you need to stretch lines like this. Similar diagrams can come in any examination of yours where you will be asked to identify. So the diagrams which are given in the NCERT on the right hand side page, all the 4 diagrams. So this way you need to show the thickening in the case of sclerenchyma. I have made a hexagon and then I am outlining it with another color so that it is prominent to you. So this is the thickening that is seen in the case of sclerenchyma. This thickening material in the case of sclerenchyma is lignin. And the more the lignin is there in a plant, that means more will be the mechanical strength. So sclerenchyma exists in 2 forms. One is the cellular form and the another is the fiber form. Let me draw it again. So in the case of fiber form, make sure that spindle shaped structure should be visible. Means this will be broader in the center but narrower at anterior and posterior end. And the central space, this central space in the case of a fiber, right? This central space is known as lumen. And this lumen is blocked because of the lignin. So these are the sclerids that I have drawn. It exists in two forms. One sclerids and the other is fiber. So this is one, this is two. This is how sclerenchyma representation should be there. You might have read about jute fibers, hemp. These are all the fibers that are there in the plant and they are actually sclerenchyma. So these are economical fibers. You might have read about them in class 8th as well because there was a chapter in class 8th, fiber to fabric, I suppose. So we are done with meristematic tissues. We have drawn apical meristematic tissue. We have drawn lateral meristematic tissue, intercalary meristematic tissue. And in the simple plant tissues, we are done with parenchyma, collenchyma, and sclerenchyma. In case students, you have understood the concept, please do hit the like and you can share it with your friends so that they can also understand it well. A playlist of fundamental unit of life has already been uploaded. The difficult diagrams of fundamental unit of life are also being done. In case you are interested, you can come to the playlist section. So we are coming to complex permanent tissues now. So in the plant tissues, the last part is complex permanent tissues and it includes xylem and phloem. Before we go ahead, the subparts of xylem and phloem should also be known. So, xylem is made up of tracheids, vessels, xylem fibers, and xylem parenchyma. So, these are all the components of xylem, and xylem helps in the conduction of water and minerals. So, if I see for all these four parts, part number one, tracheids, two, vessels, xylem fibers. What is the peculiarity in them? That they all are dead. Dead means their lumen is blocked. Why their lumen is blocked? Because they have lignin in them. So, in case a plant has more xylem than phloem, that means the plant has more mechanical strength in it. Okay? And this part of the plant, xylem parenchyma, it is the living part. Now, if I talk about phloem, it is made up of sieve tubes, sieve cells, companion cells. Okay, I'm just writing C for it. Phloem fibers and P for phloem, phloem parenchyma. So five components makes up the phloem and phloem helps in the transport of food. Right? So you should know and learn these parts first so that you can do the labeling of all these parts while drawing the diagram. So let's go ahead and draw the diagram of xylem and its components first. So a tracheate is drawn like this. In your book, there is a point being written. So this is in the pits part, a part has been labeled as pit. I will be making it right now. So this is how a tracheid look like. And inside the tracheid, we have pits. Pits are the regions where thickening is less. So in the exam, you should definitely know how to draw because in certain schools, the diagrams are also being given. Now we are coming to the second part, which is the vessel. So a vessel has numerous pits on it. So pits are the areas where the thickening material will be less. In case we are talking about thickening, here the thickening would be lignin because tracheids and vessels, they both are the dead components of a plant. So the more their number is, the more will be the mechanical strength in a plant. So this way you can make the pits, and don't forget to label once you are done with the diagram. So always while making a diagram, you should definitely label it at the end. Let me make xylem parenchyma here only in between. So in case you are making the xylem parenchyma, because this is the living component, that is why I have used the green color. So in the plant, we will have all the four components with few exceptions in gymnosperms. Again, we'll be making the second layer like this. Okay. So this is going to be the green part that I have drawn for you right now. This is going to be xylem parenchyma. I hope you are clear with the fibers that were being drawn in sclerenchyma. The same fibers can be drawn for xylem fibers. Okay, so that's why I'm not drawing that. So first part is trachete, then vessels, then third part that I have drawn xylem parenchyma, and the similar diagram that was drawn in the case of fibers of sclerenchyma, same diagram can be drawn for xylem fibers. So trachete, vessels, and xylem fibers are dead because their lumen is blocked by lignin, whereas xylem parenchyma is the living. Xylem helps in the transport of water and minerals from root to the shoot. So the source of water is root, and the sink. Sink means where it is used. So in case of transport, you should know source and sink. In the case of xylem transport, root is the source of water and sink is the shoot because it is just using the water. So we are done with the diagram of xylem as well. Now let's come to the phloem, and in one diagram only we will be doing all the labels together. So I'm drawing companion cell right now. This is its nucleus. This is one companion cell. This is another companion cell for you. Companion cells, they have got nucleus in them. This is the sieve plate that I am drawing it with black. Plate means the wall at the end. If you could see that I am making gaps in between because these gaps will allow the passage of the food from up to down or from down to up. So this is going to be my sieve tube. This is going to be the sieve plate, and just adhering बिल्कुल पास में इसके होगा companion cell, and just close to this we will be having phloem parenchyma. The way we have drawn xylem parenchyma, similarly we can draw phloem parenchyma. Sieve tube and companion cells, they are siblings, right? Means they are formed from the same mother cell. But sieve tube is a one that is not having nucleus of its own. So there is no nucleus in it. So all the nucleus related activities of this cell are being taken care of by companion cells. That is why on either side of the sieve tube, what do you have? Companion cells. What are these? These are phloem parenchyma. Similarly, we can use the same fiber diagram that we have drawn in sclerenchyma for drawing the fibers. So phloem, it helps in the transport of food. It does the transport in both the direction, means from down to up and up to down. As xylem was only doing in one direction, it was unidirectional transport, but phloem does bi-directional transport. So with this, we are done with the diagram of phloem as well. Now, in certain schools, protective tissues are also being taught. Protective tissues are a part of plant tissues only. So in this, epidermis comes. So let me quickly wind it up with these also. Epidermis is the outermost layer of root, stem, or leaf. What is it made up of? So it is made up of compactly arranged parenchyma cells. So when the parenchyma cells, they arrange compactly with no intercellular space between them, then they form the epidermis. Another protective tissue that you should know is cuticle. You know, in desert, there is scarcity of water. So in case a plant is going to lose so much water, then a plant will become deficient. So to prevent the loss of transpiration, just on outer side of the epidermis, there is an additional layer being present, which is known as cuticle. So cuticle is the one that I have made with blue. Epidermis is the one that I have made it with black. So cuticle will be the outermost layer in the desert plants, which will prevent the rate of, which will slower down, and it will prevent the rate of transpiration. So all the plants do not show this. And cuticle contains a chemical substance in it, which is cutin. This is an important MCQ question for class 9th. Students, in case you are liking this explanatory video of learning the diagrams, please do hit the like button. There are some more explanatory videos for you in the playlist. In case you are interested, you can check it out. Uh, Next one, in protective tissues, right now we have seen epidermis, we have seen cuticle. Now let's see about cork. This was the same cork that was being used by Robert Hooke to discover the cell. So to draw the diagram of cells, it is very easy. You can simply draw some flat cells like this, and you need to overlap another cells on it as well. So it is just having, each cell is having nucleus like this. So what are these? These are the cork cells, and cork is only found in the dicots. In case you have not heard about it, you can replay this video again, because I've already explained earlier that what are dicot plants. So this is how you should make the cork. And you know, cork also has a chemical substance which is known as suberin. So suberin is impervious to water, and it is impervious to gases. So this is another protective tissue that you need to study in class 9. Also students, the epidermis that we have done earlier, it's not regular. In the epidermis, we have stomata also. So what are stomata? So the next heading is going to be stomata. These are tiny pores on leaves. Generally, their number is more on the leaves. And what are their two functions? They help in the gaseous exchange, means the exchange of the gases, because for photosynthesis, CO2 should go in, and the oxygen that is being formed should come out and is used by us. And the another function of stomata is that it helps in transpiration. Transpiration is the loss of water in the form of water vapor from the aerial parts of the plant. So as we have understood what is stomata, let's learn its diagram. So for making the diagram of stomata, make two kidney structures like these, and they will be having chloroplast in between. So these green circles are the chloroplast, and this bigger kidney-like structure, this is guard cell. So the guard cells are surrounded by these red marked cells, and these are known as subsidiary cells. Subsidiary cells. These green cells are guard cells. The space that is formed in between this one, this is stomatal pore. Because of this pore, water comes out and the exchange of gases takes place. Okay, this is the chloroplast which is present. So the part that I'm coloring now is the chloroplast, and this black part, black circle, is the nucleus that is found in the guard cell. So in stomata, a stomata is made up of two guard cells, a stomatal pore, and few subsidiary cells. So epidermis is not regular in plants. In the case of leaves, these stomata, they are more. Before we wind up this video, let's see the thickening material in all. So you need to learn the thickening material that is there in parenchyma. Parenchyma is a simple permanent tissue, and so as collenchyma is, along with sclerenchyma. So all these three are simple permanent tissues. So the thickening material in the case of parenchyma is cellulose. The thickening material in the case of collenchyma is pectocellulose. The thickening material in the case of sclerenchyma is lignin. The thickening material in the case of cuticle, in case you have joined it late, I have explained it. You can replay the video. Cuticle contains cutin. And in the cork, what do we have? We have suberin. All these thickening materials are to be learned thoroughly. NCERT exemplar also has the questions on it, and even these questions can be given to you in the MCQ examination. This is all for today. In case you have understood the video, do hit the like button, and you can subscribe if it was helpful to you. Stay connected. Thank you.